Next, I'd like to introduce Callie Concannon, who's a master's student at the University of Connecticut with me, and she will be talking about her studies on ocean acidification impacts for the Atlantic Silverside.
There we go. Okay, so my name is Callie Concannon, and as Hallie was saying, I'm a master's student at UConn Avery Point in Hannes Bauman's lab, and I study how increased ocean temperatures and ocean acidification together impact growth and reproductive output in the Atlantic Silverside. So a little bit about the Silverside first. They're pretty small coastal fish, and they're super abundant in coastal areas from Nova Scotia down to northern Florida, and they're a common bait fish. They feed on smaller organisms like copepods and brine shrimp, and then they're subsequently fed on by larger sports fish like striped bass. And they became an important model species to study climate change impacts on coastal fish because they are small and easy to catch, and they're super abundant close to home, and they're easy to raise in captivity. And they're also an annual fish, so their lifespan is generally 1 year, sometimes 2 tops, which makes studying long-term exposures a lot easier. And as Stephen was saying, coastal areas are very dynamic. So a lot of what climate models are projecting for increased temperature and pH for the upcoming years, a lot of coastal areas are already experiencing this. So this is a time series across 5 years in Mumford Cove, which is where we collect our silversides in Groton, Connecticut. And I have average sea temperature in red, dissolved oxygen in blue, and then pH in green. And the area I have highlighted here is the silverside spawning season. So we're seeing that at the beginning of the silverside spawning season, the pH is relatively high and temperature is relatively low, and it's less variable. But as you move towards the end of their spawning season, there's a lot more variability, and the pH has dropped and the temperature starts to increase. And we're seeing pHs that are in these projection models. So we're seeing pHs as low as 7.5 that they're experiencing, at least on it for daily time scale, but even some seasonal fluctuations. And silversides are batch spawners, so they spawn every 2 weeks in here. And so even though they're experiencing these conditions already, they're predicted to experience these conditions more frequently as time goes on. So how would these changes over a long-term exposure impact the local fish? And most of the research done on silversides already have focused on shorter timescales, predominantly their early life stages from eggs, larvae, and juveniles. But there's a general knowledge gap in long-term exposure. So if they're exposed growing up in a low pH, high temperature environment from an egg to a spawning adult, how does that impact the species, which is the question I aim to address. And there's also a knowledge gap in how these long-term exposures impact the amount of offspring that the species can produce. I don't know if you guys can hear this in the background, but one of my neighbors is moving, so if there's any loud bangs, it's them. So the question I aimed to address was how long-term low pH, high temperature environments, being in these environments from egg to spawning adult, impacts both their growth and then also their reproductive output. So how many eggs are they producing and what sizes are they? And past research on shorter timescales have shown that silversides are relatively tolerant to these exposures. And given that these aren't uncommon values for them to experience, we anticipated that there would be no effect on their growth and reproduction. So how do we study the silverside? First, we capture wild silversides using a sanding net seen on the left. We then bring them back to the lab and artificially spawn them and then raise them in tanks seen on the right under a variety of temperature and pH conditions. So my conditions are in the table below. The temperatures I picked were 17 and 24 degrees Celsius, and the pHs were 7.5 and 8.2. So 17 degrees Celsius and a pH of 8.2 are like the beginning of their spawning season when things are less variable, and then the 24 and 7.5 are the end of their spawning season. What projection models are predicting will become more frequent for them to experience. So I raised them in these conditions for 11 months. And then after 11 months, I measured their total length, which is their length from their snout to the tip of their tail, their weight, and a gonadosomatic index, which is the percent of their body weight that is their reproductive organs. And I also took a deeper dive into the female reproduction and looked at how many eggs they're producing, what sizes those eggs are, and what developmental stages those eggs are in. So I have some pictures of some fish eggs here. So I counted and measured all the eggs in the ovaries for the females, and being a batch spawner, you see a wide range of sizes. So being batch spawners, you have this big peak in very small egg sizes, and then you see another peak later on of like larger sizes, and there's a wide range in between of all these different sizes, because batch spawners, technically they like take from that smaller group, start growing up a group of eggs, and then spawn it, and then 2 weeks later redo the same thing. So I looked at both the maximum size of the eggs, the minimum size of the eggs, the overall range, if there was any difference in those ranges across these tank treatments. And then developmentally, I looked at what developmental stages these eggs were in, and if there was an effect on what sizes the eggs were in those stages, and also if they were reaching the same developmental stages. So as I said before, we anticipated that growing up in these high temperature, low pH environments would result in no effect, but we actually are seeing some significant effects. So in these warmer, more acidic environments, we're seeing that the females and males are growing larger and heavier, but their gonadosomatic indexes are smaller. The females are also producing a lot more eggs, but they're on average of a smaller size. I'm still analyzing those egg size ranges, but it looks like there may be a small shift, but if it's significant or not, I'm unsure of right now. And then developmentally, they're all reaching the same developmental stages in their ovaries. All the eggs are reaching the appropriate stages, and they seem to be around the same size, so that's not being affected by the temperature and pH. Overall, there's lots of variability in this study, but there are some clear pH and temperature effects that we're seeing. And I just want to thank my lab group for helping me out through all of this, because there was a lot of fish over a year to take care of.
Awesome, thanks, Callie. And last but not least, we have Dr. Matthew Sasaki, who is at UConn with me and Callie and just defended his PhD on Monday. So congrats. He'll be talking about thermal adaptation in copepods.
There we go. Now I'm unmuted. Awesome. Thanks, Callie. And Hallie for the introduction. So yeah, like Hallie mentioned, I just finished my PhD in oceanography over at UConn. But my background is in evolutionary biology. And so the main questions in my research aren't directly related to, or aren't directly focused on vulnerability to climate change. But one of the things that I think is really important is that we remember or that we think about how evolutionary dynamics might influence vulnerability to climate change. And so that's what I'm going to talk about today a little bit, which essentially boils down to this question of why do plankton do what they do? And just to make sure we're all on the same page, I'll remind people what plankton are, right? Plankton is a really diverse group of organisms that are defined by how they live, right? The fact that they live suspended in the water column. And we care about plankton because things like phytoplankton form the base of the food webs, which provide energy for animals like zooplankton, and then eventually animals like fish. But one of the things that I like to point out is just the fact that plankton are so diverse, right? We tend to lump them into these groups like phytoplankton and zooplankton, but it's eye-opening to put a water sample sort of under the microscope and just look at how many different things are living in the water that you wouldn't normally be able to see, right? Most of these animals are less than half the size of a grain of rice. So I could spend all day talking about plankton and how cool they are, but I'll stop and I'll introduce the animals that I work with, right? I work with one particular group of planktonic organisms called copepods. There's a illustration of just a couple of the more diverse types of copepods shown here. But yeah, so the main thing about copepods is they're super diverse, they're super abundant, right? Copepods are the most abundant animals in the ocean, if not on the entire planet. And I think that they're just fascinating animals to study. They're also important animals to study. The reason why we spend time and effort studying copepods is because they are such an important food source for fish, right? Callie mentioned that silversides eat copepods and then pass that energy up to larger fish. But copepods are also a direct food source for things like whales, right? In the cooler, high latitude waters, copepods get bigger and can be a really important food source for things like North Atlantic right whales. The other reason copepods are super important for the way the ocean works is because they're so abundant, because there are so many copepods in the ocean, they actually play a role in how elements cycle through marine systems, right? Copepods take carbon from phytoplankton, package it into these torpedo-like hydrodynamic fecal pellets, which can carry carbon from the surface waters to deep waters, right, where it's sequestered for long periods of time. I am working with copepods as part of my, I worked with copepods as part of my PhD because they're so widely distributed. And this is important for me because my research tends to take on two broad areas, asking how organisms do what they do by looking at the relationships between DNA, the genes that they encode, and how those genes eventually lead to differences in how organisms function. The other question that I tend to ask that my research focuses on is why organisms do what they do, and this is where evolution comes in. Right, large-scale patterns in the environment, either over spatial scales or temporal scales like seasonality, those patterns in the environment shape patterns in adaptation, right, over evolutionary timescales. And ultimately, evolution, in my mind, is why organisms do what they do, right? The process of natural selection over time shapes what organisms do. And so copepods are an awesome model system to study these two questions in because they're so abundant, they're super widely distributed, and they have a really short generation time, right? Just less than a month in most cases. Okay, the last thing that I want to touch on before I highlight a little bit of my work has to do with genetics. To an evolutionary biologist, in many ways, organisms are just vessels for genes, right? We all carry around our DNA, and at some point, the goal is to replicate your genes for the next generation. And this is actually really important to think about when we're dealing with planktonic organisms because they're being carried around by currents, right? This is from a really cool simulation from NASA about how currents move through the ocean. And all of this water movement as it moves throughout the oceans is carrying planktonic organisms in the water, right? And as those organisms are moved around, their genes are being moved around as well. And because the genes have such a strong impact on what organisms do, when we're looking at patterns in adaptation, gene flow is also an important thing to consider, right? So how these genes are being moved around is also going to shape patterns in what we see organisms doing. So those are kind of the central themes for my research. There's a couple of projects that I've worked on that I want to highlight. As an undergrad, I worked with this little tiny copepod that lives in tide pools called Tigriopus, looking at patterns in thermal adaptation. I spent some time working with Antarctic invertebrates doing sort of reduced genome sequencing to look at patterns in gene flow and so how connected are sites across the Southern Ocean. And then all of that kind of gets combined in what was my dissertation project, looking at this copepod Acacia in the North Atlantic. So I don't have too much data to show, but one of the things that I did for my dissertation was go out and collect these live animals from the Gulf of Mexico up the Atlantic and then into Canadian waters. So that covers this really large temperature gradient, right? So these animals are found across crazy different environments. We were able to bring them back to the lab and then measure things like thermal tolerance, which is essentially the temperature, the upper limit, right? The upper temperature that organisms could handle living at. And then I was able to compare thermal tolerance between populations. So that's what we see here, is that there's some large variation between populations across this environmental gradient. We also found evidence for gene flow, right? These are populations that don't have different thermal tolerances. We did some genetics that showed that there's enough connectivity to keep, to essentially prevent adaptive divergence from happening. One of the other elements of this project was comparing male and female thermal tolerance. And we actually found that males always have lower thermal tolerance than females, which has interesting consequences for things like vulnerability to climate change. And then the other main aspect of my dissertation that I want to touch on is seasonal variation, right? So Callie mentioned how seasonal Long Island Sound is, right? Specifically Mumford Cove, which is coincidentally where I was collecting copepods for my entire dissertation, for most of my dissertation. And so I was able to piggyback off of that environmental data set that she showed and use that to look at how copepods in Long Island Sound respond to that seasonal variation. With silversides and sort of their generation time of the year, they experience that entire range of environmental conditions within a generation. Copepods don't do that, right? With a generation time of only a month, now they experience those range of temperatures between generations. And so that means that there's the potential for evolutionary dynamics to kind of take over when we're looking at what copepods are doing throughout this annual temperature cycle. And so I know that the graphs are a lot. And so the main two take-home messages and how I've related my work on sort of the evolution of thermal tolerance to vulnerability to climate change. One of the things that we found is that spatial patterns in vulnerability are going to be affected by things like patterns in selection, right? And patterns in gene flow, which are important to consider when we're thinking about sort of big picture vulnerability. The other main take-home point that relates to vulnerability to climate change is that seasonal variation in temperature and other environmental factors could be an evolutionary force that creates and maintains adaptive genetic variation. And so it essentially primes populations, or it might help populations respond to new conditions by maintaining a lot of diversity within a population. And so I'll end there. There's a bunch of people I want to thank. A lot of my work was funded through our department, UConn Marine Sciences, as well as grant money from NSF and Sea Grant.
Thanks for watching. For more information, visit our website at www.futurefrogmen.org.